Halo, selamat datang di acara resital musik klasik virtual DIPI 2022 yang diselenggarakan untuk menggalang dana penelitian ilmu dasar. Selain menyelenggarakan pagelaran musik, kami juga akan bertemu dengan para ilmuwan yang memiliki reputasi internasional dan kita akan berbincang-bincang, kita akan mendengarkan perbincangan dengan salah satu di antaranya pada hari ini, yaitu pemenang hadiah Nobel bidang kimia dari Amerika Serikat, Profesor Martin Chalfie. Prof. Martin, how are you? Good morning.
I'm fine. I'm happy to be with you.
It's an honor to meet you virtually, Prof. So I'm here with Prof. Retno as well.
Hello, Mbak Vika. How are you?
I'm good.
I'm fine. And you? Good.
And hello, Marty. How are you?
I'm very good. It's a beautiful day here. I hope you're having as nice a day in Indonesia.
We are dark now. Thank you very much, Marty, that you are willing to join us.
It's late. It's almost time for bed. Sorry.
No, it's okay. I'm used to do it since this COVID pandemic.
So before we start our conversation, let me introduce our special guest for today. Jadi Profesor Martin Chalfie lahir pada tanggal 15 Januari 1947. Beliau adalah ilmuwan Amerika yang bekerja di Columbia University. Beliau menemukan Green Fluorescent Protein atau GFP yang dapat digunakan sebagai biological reporter. Dan beliau memenangkan hadiah E.B. Wilson Medal di tahun 2008 dan hadiah Nobel untuk kimia pada tahun 2008 bersama dengan Osamu Shimomura dan Roger Y. Sien. Kemudian narasumber kedua kita, Prof. Retno, yang sudah tidak asing lagi. Beliau adalah ahli penyakit jamur yang mendapat gelar guru besar dari dua universitas sekaligus, yaitu UI dan UKI. Dan sekarang Prof. Retno fokus untuk meneliti jamur histoplasma yang dicurigai endemik di Indonesia. Dan selain berprofesi sebagai pendidik, klinisi, dan peneliti, beliau juga gemar bermusik dan merupakan seorang penyanyi sopran musik klasik, dan kegemarannya terhadap musik klasik lah yang mendorong beliau untuk menginisiasi acara resital musik klasik DIPI kali ini. So, I will ask you my first question, Prof. Martin. What inspired you to become a scientist?
So different things inspired me at different times. When I was growing up, there was a series of books, and my mother kept them, so I can actually show you these books. These books were called the All About books. So this one is all about dinosaurs, and then there was another one all about atoms. And I love these books. I read them all. So when I was a kid, I was sort of fascinated by this. There was also a wonderful, wonderful television program about blood and how blood worked. And the major protein in blood is hemoglobin. And this television show in the 1950s was called Hemo the Magnificent. And
Hemo the
Magnificent. About the blood, but it was very impressive. And I don't know if that was why I got interested in biology, but that was a start. Then I went to school. I was fairly good in sciences and math. And then a horrible thing happened. I worked in a laboratory and everything failed. Everything. I could not do anything right. And I decided I'm not going to be a scientist. This is not what I should do because I am a failure at it. So I did other things and then found myself several years after college needing to get a summer job. And a friend told me about a laboratory that I could work in. And I interviewed and I went to work in that laboratory and the experiments worked. Not only that, I had an idea that worked. And I was so buoyed by the idea that maybe I could actually come up with ideas. And it was fun when the experiments worked, at least. maybe this was something I should try out again. So I like science, and I decided I wasn't going to do it. And then I got back into it because an experiment worked for me.
Okay, that's very nice story. Can I ask you another question, Marty? You did an amazing work, and you have a long-standing reputation on researching a very specific protein. That is the famous and very well-known GFP. Would you please tell us a little bit about this GFP? And I would like to know what are the reasons for choosing that specific protein? Why not DNA, which it is now is very fashionable? Please, Marty.
So it has to do with so GFP is, let me explain what GFP is. GFP is a protein that was first found in a jellyfish. And it has a very unusual property. Now, there are many organisms, fireflies, glowworms, bacteria, fungi, and fish, all sorts of animals that can generate light. And so scientists like Osamu Shimomura were looking at the biochemistry of how is light produced? How does an organism generate light from chemical energy? And in the course of studying this, he discovered a protein that had a property that no one thought of. People knew that there were proteins that could cause the chemical reaction to make light, but he found a protein that changed the light's color, absorbing blue and ultraviolet light and then giving green light out. And this is what we call green fluorescent protein. It was the very first fluorescent protein. And fluorescent protein, again, is one that takes in light of one color and gives off light of another color. Now, he did this work in 1962. I didn't know anything about it until I heard a talk in my department by someone who was looking at a related organism that also had GFP in it. What was I doing at the time? At the time, I was interested, as I still am, in how nerve cells develop and how particular types of nerve cells work, particularly cells that sense mechanical signals like touch in our case. And I am a geneticist, so we were finding mutant animals that could not sense touch. Well, after you find the mutants these days, you clone the gene and ask one initial question. Where is this gene active? Okay. Now, there's lots of ways of doing that. You can say, you know that the DNA makes RNA, so you can look for where is the RNA. And people have methods of doing that. Or you can say, well, the RNA then makes protein. Let's use an antibody and find out where the protein is. Okay. But all of those methods, and they all worked and they all gave us the right answer, are methods or were methods in which you had to really prepare the samples. And by prepare, I mean fix them so they were dead. They weren't dynamic. They were just what you prepared at the moment, because you then had to poke holes in it to get the reagents in to be able to see where these things were. And as I listened to this talk about GFP, a protein that you didn't have to add anything. All you had to do was add light. You didn't have to add any chemicals. So you didn't have to poke holes in the sample. I realized this is something, if we could put it in our organism, if we could put it in our cells, we would be able to see where the genes were active, simply because this would make GFP instead of the normal product. And that way we could see this and look in living animals. And so GFP was just a really wonderful way of looking at things. So for example, I worked on this very tiny 1 millimeter long worm. It only has 302 nerve cells.
Okay.
And I study 6 of these 302 nerve cells. But if I look in the animal, I can't see them. But if I arrange to have GFP in those 6 cells, then I can look in a microscope and they will fluoresce. If I shine blue light on them, I will see them as green, and I will see them in the living animal. This means I can watch them grow, and I can now start to look for mutants that might be defective, not have enough cells, have the cells in the wrong place, have the cells growing differently. And all of those lead to fascinating problems in biology. So the 2 main things about GFP are, 1, it doesn't need to have anything but light to make it green. And 2, and for that reason, you can use it in living organisms. It also has another couple of features. It is a monomer, that is, it's only 1 molecule, not many, and it's small, so it goes everywhere in the cell. So you see the whole cell, not just a small part. All of these things said to me when I first heard the seminar, this will be a terrific tool. And that's why I got interested in it.
Okay.
And it's proved to be that.
Yeah. Okay, it's quite interesting then. It's very, very, I just curious how to do that in my field in fungal diseases, Marty. One day I will discuss it with you. Okay, thank you very much. Can we move to the next question, please? Marty, as a researcher, I'm sure we need independent financial support to do our work. Could you tell us a little bit about how to get that kind of research grants, independent one? And does that grants allow you to do research freely? Also, what is the role of your university in ensuring that you can conduct your research freely? Please, Marty.
So I feel that at least for my research, it was greatly aided by both of those things, the ability to get grants and the freedom I have to choose what research I want to do at my university.
Okay.
Now, I'm not sure I'm the right person to say how to get a grant. That's always difficult, and I don't always succeed any more than anyone else does. And, you know, everybody is competing for money. The actual writing of grants entails having often experimental ideas that are very focused, and you understand what you're going to do, and you can convince other people that there is something that's going to come out of that that's very important. But it's still difficult because we have way too many ideas and way too many good ideas than there is funding. So it doesn't always happen. But there's a big difference, at least here in the United States, I hope elsewhere, between what we know of as grant support and what most business people know of as contracts. A contract, which the business world is full of, a contract says, I promise to do the following 3 things, 3 experiments, develop 3 things or whatever. And when I do those 3 things, you will pay me. So we're promising to do, in a contract, you promise to do specific things. Science doesn't work like that, in my opinion. Because in science, what if somebody makes a discovery the day after you get funded that changes everything? How you think. What if you make a discovery that changes everything? Do you go back and do what you said you did in the grant proposal when it's already done? Or maybe there's something more important to do? No, and I don't think the funding agencies think that either. What they're doing in a grant, by funding a grant proposal, is they're saying, we believe in how you are doing your science, and we want to support that. Your ideas sound good to us. But, and this is the part they never write, they'll never admit this, but it's really true. If you come up with a better idea, if you can think of something that will push the science faster and further than you said in your proposal, go ahead and do it. Who's going to stop you? You have the funds. Use the funds in the best way. Now, in a couple of years, you're going to have to renew that proposal. So you have to show that you've done something. But we will listen to you. And GFP is a very good example of this. I could never have written a grant proposing to look at GFP because people would have said, how do you know what's going to work? How do you know what's going to happen? What do you're not showing us any beginnings to these experiments. You just have an idea. And so I didn't ask for any money. We just did the experiment as part of what we were doing. And I think that that's what we were expected to do. We were expected to go and try the unusual, to try the chancy thing and see if it worked. Because if it did, that would be wonderful. In the case of GFP, it did work. We put it in the bacteria and we saw them glow green when we shone blue light on them. So that idea of doing it, I should say, I've been funded in the United States by the National Institutes of Health. Osamu Shimomura was funded by the National Science Foundation. But his discovery of GFP and my showing that GFP could be a biological marker, they were in no grants, the things that we were doing. But both of these agencies went to the federal government and said, you want to see proof that we're doing good things? Look at GFP. They both used it. And I think they were right to use it as an example, not because we wrote grants about it, but because their support helped us do those important experiments. So I think that's the idea about grants. The idea about university freedom. My university doesn't tell me what to work on. I would not expect it to. I would not want it to.
Okay.
But they are supportive of me doing my experiments. They give me a lab. They allow me access to students, colleagues. They give me an environment where I can do research. Would I like that environment always to be better? Would I like more resources? Yes, I think we all would. But I would have as a distinction university freedom to do research, and especially the type I do, which is basic research, and working in a company. There is nothing wrong with working in a company, but companies have very specific goals.
Yeah.
And if the project is not working the way they think it should, and they think there's a better thing, you can't say, oh no, but I like my experiments, I'm going to go do them. You're working in a company, you have to be part of that operation. And so there's some wonderful resources and great freedom to a certain extent, but if the project is dropped, that's the end of the project. I have a little more flexibility. And there's nobody deciding, nah, that's not an important experiment, that this is not an important experiment. I have to make that decision myself.
Okay.
Very nice. Okay, Mbak Vika, please.
Yeah, Prof. Martin, in your opinion, what are the long-term implications of research for humanity and industry in general?
So I think we have some terrific examples of this. The COVID-19 vaccines, they were produced in an astonishingly short amount of time.
Yeah.
But the basic science underlying them was decades in the making.
Yeah.
If we had not had the science, we would not have had the vaccines. We would not have been able to treat these diseases in the way we do. We can look, there's a company in the United States, I think, called Quartzoft. And in 2014, they made an infographic that a friend of mine told me about. Now I show it in my seminars. This infographic is this enormously long poster. You cannot see, you got to really magnify it to see the parts to it. But at the very top of that poster is the Apple iPhone.
Oh.
Okay.
And there are lines that come from the iPhone to all the scientific discoveries that were needed for that single piece of equipment to be made. And you go back to the early 20th century, early 21st century, early 20th century, you keep going down and down the chart, and there are hundreds of discoveries that are needed for the Wi-Fi, for the mathematics, for the lens and the camera, for all of the things that are in this one little contraption. And then at the very bottom, you get the first lens, the discovery of binary, the discovery of zero, all of the things, all of the science and mathematics, physics, the chemistry, everything that is needed to actually culminate in a product. The American author, Mark Twain, in a letter that I found from 1903, was writing, in this letter, he said, you know, it takes hundreds of people to make a phonograph or a photograph or a steam engine, or the things he was talking about in the 1900s, early 1900s. He said, it takes hundreds of people, but we only know the last one. We only know that last person that did it. But there's so much that's needed as the groundwork that is needed to be able to get advances in medicine, products that help us in our daily lives. All of the things that come out of research are the result of lots and lots of activities by lots of people. And many times we don't know what is going to actually happen, what's going to be the important thing that's going to do it. Let me give you one example, because I really like this example. I can't tell you the names of the people that did it, but there were some researchers that were studying a breed of rats. And when they looked at these rats, they were making a very special breed. And so they wanted to be very careful with it, really coddle the babies when they were born. And so they took the baby rats away from the mothers to make sure they were in the right environment, nothing was going to happen to them. The baby rats did not do well at all. And this puzzled the investigators. And so they looked at the rats and they said, maybe we should look at what the mothers do when the rats are newborn, when the babies are newborn. And what they found was that the mother rats were licking their babies.
Okay.
And so the experimenters decided to test something. They didn't lick the babies, but what they did do is they took a little swab and they stroked the babies. They touched them.
Okay.
And
Yes.
when they did this, touching the babies did really well. Now, so they realized that the touch of the mother was very important for the babies. Okay, is that a result? Is that something about? Turns out that as people have now looked at premature babies in hospitals, they do much better if they are also stroked and touched and stimulated. So this led to an understanding of what we should be doing to our most vulnerable newborns and how to help that. So there's an example. If you never know where it's going to come from.
Okay, thank you, Marty. Let me continue with the next question. Marty, I believe that regeneration is very important in any field of research, so that the young will continue our work. What do you think about that? Do you agree with me? If so, what is your plan for the regeneration? Please, Marty.
So I think, so if you ask me this, there's an answer I would give you all the way through my career. And there's an answer, I think, as I'm now much older, I'm thinking about in a little different way as well. But you're absolutely right. Teaching is an education, and developing the next set of investigators is exceptionally important. The way we do this in my lab is I often take, this summer, I have 4 undergraduates working in the lab. Working on projects, so they get some experience working in the lab. I have talked in high schools, I've talked in elementary schools, get people excited about science. I think that we do need to communicate our excitement and our enthusiasm about science to as many people as possible. But it's not enough to talk about why we like science so much, or to encourage people to do it. We have to give people the opportunity to do science. Okay. I'm connected with a group called Society for Science. They put out a journal called Science News, but they also sponsor what's called the International Science and Engineering Fair, ISEF. And this is a science fair that high school students from all over the world compete in. They come, it culminates science fairs all, and these high school students are astonishing. Yeah. What they have, their ideas, what they are doing, what their enthusiasm is, all of this is wonderful. So regeneration is absolutely required. We have to educate, stimulate, encourage young people as much as possible. But we also have to enable them. We want as many voices as we can in the scientific community, because everyone brings a different perspective. And the idea that there is a hierarchy in science, I think, is one of the most destructive, that somehow there's the big professor on the top and all the other people doing just what the big professor does. I can tell you that we would not have done most of the things we do in my lab if that was what is going on. It's a real sharing and interaction among people that I think really promotes science.
Prof. Martin, if you can say anything to your younger self as a scientist, what would you say? And what is your message for the younger scientists today?
Maybe a little bit different. My overall message to myself is, don't be an idiot. But I think that I wouldn't say that to other people. To myself, to be really honest, I'm not sure there is really good advice to give anyone, or that anyone really takes any advice, because I don't think I would have listened to anybody when I was growing up. And maybe that's a good thing, maybe it's a bad thing, but I know that almost everybody thinks to themselves, oh, if somebody had just told me to do X or to do Y, my life would be so much better. That doesn't happen, I think. I think the idea is, I think what I would, if there's anything I would have really liked to have told myself, is go more deeply into things. Really learn about the stuff. Become enthusiastic. Ask yourself questions. Always ask, what's the next thing to do? What are, think about the things. When we start out in a laboratory, and I see that with undergraduates all the time, and it happened with me too, we are very concerned about training our fingers and training our eyes and training ourselves to physically do the experiment. So much so that we stop thinking about what does it mean, or what are the implications, or how should I be thinking about what's next? All of these things, I think, are very important questions. And it took me a while to realize I should be doing that, not just accepting what's going on at the moment, pushing that ahead, thinking about what I should be doing in the future, thinking about what the implications. If you hear somebody talking about something, it's not just, oh, that's nice information, but, oh, I wonder if that applies here. I wonder if that is important for me. I wonder what I can think about this. I wish I had done that more when I was younger, but as I said before, I'm not sure that I would have listened to myself. And maybe I shouldn't have anyway.
Let's talk about other things, Professor Martin. Besides your daily work in lab or your research project, what is your hobbies? I heard that you play some musical instruments.
I play classical guitar. My father was a professional guitarist. He, in the 1930s and early 1940s, he played in an orchestra, and he was, at the time, of course, there's no television. Radio stations were really big, and the big radio stations had their own orchestras, and he was the staff guitarist for one of these radio stations. And when I was about 12 years old, he said to me, because I was the oldest of his three sons, he said, okay, you're getting a guitar, but I don't want you to play this popular music. I want you to learn classical guitar. So I learned classical guitar and have loved it ever since. And so I've enjoyed it a lot. But to talk about these things more generally, the world knows me because of the science, because of the Nobel Prize. But that's not who I am. That's an important part of my life. I am very appreciative of that. But that's not what my life is. I have a family. I mentioned my daughter, my wife. We do things. We enjoy reading. I enjoy playing music. I have friends. I have a life. Science is a very important part of that life, but it's not the only thing in the life. And frankly, I don't think that is necessary. And I think everybody decides how they want to balance their lives and how much of their work and their interests, and it varies for everybody. But to me, I don't think I could be just one person, doing one thing. I have to say that the Nobel Prize has given me opportunities that I should have taken before this, but I didn't. But have sort of thrown them these opportunities at me in a way that I'm very, very grateful for. I get asked to give these interviews and to meet people in different parts of the world that I've never had a chance to meet before. And that's fascinating. The other aspect is a couple of years ago, I have several jobs now. One of my jobs came about out of the National Academy of Sciences, and that is I am the chair of their Committee on Human Rights, which works on behalf of scientists, engineers, and health professionals all over the world who have had major human rights abuses. And unfortunately, there are many. And I think that my getting the Nobel put me on made people aware of me as a possible person that could contribute to this. But since I've been on that committee, I have had this opportunity to do something that I like to joke that my wife, well, I like to say that there was a person who said to me, "You have power now." That's not true, but this is what was said. You should use that to help people. You should use that to do something. And that was my wife. But she was right. If there's any help that this Nobel can make, that I can do something that allows me to do something for somebody else, I think I should use it. And so being part of this Committee on Human Rights has been part of that giving back. And I think that many of the Nobel laureates that I have met, they felt the same way. As, okay, we've been recognized. Yes, we were very proud of our science and what we did. But if now we're going to be spokespeople, why not? Let's go do that. Let's be so that's become another part of my life that I'm very proud of, because I've been able to work with some astonishing people that are very, very dedicated to others. And it's been gratifying and very, it's been a wonderful thing. And I've made many new friends in this way. So I think our lives often take different turns and go in different directions, and we add to them. And it's not just the science. The science is an important aspect, but it's not, and maybe the way people view us, but it's not the whole thing in our lives.
Okay. Very nice explanation. Yes.
Yes, indeed. Last but not least, Prof. Martin, we would like to hear a special message from you that can inspire a lot of other scientists.
So when I sometimes give talks to undergraduates, one of the questions that comes up often is, what do you think is the next thing in the future? What is something, and what they're really asking is, what should I work on? What should I do? What's the next big thing? And of course, I don't know what the next big thing is, but it's gotten me thinking about what we do to our students. We say something to our students, at least we say something to our students here in the United States. We say, you're very talented. You have abilities. You can probably do anything you want. So follow your passion. Now, I realized if somebody had said that to me when I was in college, I would have been terrified. I would have said, I don't know what my passion is. What if I make a mistake? What if I pick the wrong thing to work on?
Yeah.
Okay.
Well, one never knows what the right thing is or the wrong thing is.
True.
And there is no one path. So if anything, I would say there is no one way. I can't tell people what they will do in the future. I can only say that people should try to enjoy what they're doing and be amazed at all the surprises that are going to come up in the future, because there are going to be amazing things, because This moment, and always the present moment, is the golden age of science. So thank you.
That's very inspiring, Professor Martin. Thank you so much for your time.
It's a pleasure.
Thank you, Marty. Thank you very much.
Baiklah pemirsa, demikian bincang-bincang kami bersama dengan Profesor Martin Chalfie, dan nantikan obrolan kami bersama dengan tamu-tamu istimewa lainnya hanya di acara resital musik klasik virtual DIPI 2022.